I wish first to express my very deep gratitude for this most prestigious award. It is like finding oneself on the top of the highest peak of the Himalayan chain, the Everest. And to be there, to find yourself there, without having had any effort in the ascent, but enjoyed a tremendous pleasure for the panorama which we saw at every turn of the road. But I must also say that I would never have had this tremendous privilege which I hold today to the Nobel Committee, were it not for a large number of events which I received, which for fortunate event, for which I have no merit, but just it was simply good luck. And I wish before starting to enumerate the one which I consider most important, even before that I got the work which brought me here in Stockholm. The first of this event was to be a student in my native country, Italy, the University of Turin, and being a student of one of the most extraordinary human beings as a scientist and a person. His name is not related to me, is Professor Giuseppe Levi. All his students were infected by his tremendous enthusiasm, by his dedication, by his profound knowledge, and by his ethical behavior, which he showed courageously during the Second War, fortunately escaped death. All of us were admiring him, all of us owe very much to him, and particularly three of us had the great honor of being here and receiving the Nobel Prize. And we were at the bench together, we worked together for 6 years, and the name many of you may know, Salvador Luria in virology, my friend and schoolmate really every day and every month for 6 years, Renato Rubecco, discovered of oncogene, or at least the problem as the field which brought to the oncogene, and finally, I'm the third of this group of fortunate students of Giuseppe Levi. The second very good event, a really fortunate event of my life, and I have a debt of gratitude for it, it was to meet Professor Victor Hamburger, an outstanding, I must say, leader in experimental neurobiology, who invited me at the end of the war in 1947 to join him for studying together the effect of the periphery on the center. Dr. Victor Hamburger, to whom I sent today all the feeling, my gratitude and my thanks, has been not only a very, very intelligent and learned scientist, but a lifelong friend with whom I shared the emotion of the beginning of his work and to whom I owe very, very much. Number 3 of my good luck has a name, and the name is Dr. Stanley Cohen. Dr. Stanley Cohen came in 1952 when I was still in Rio de Janeiro and joined me, and I believe that is far more than I am responsible for the work which followed. I admire Stanley, I must say so, not only as a very, very good scientist, but for his humility and way of living, very much, which I believe is equally important as being a good scientist, is to be a good human being, honest, humble, and exceedingly good. So it's to Stanley that I give today my thanks as I gave to the others. Finally, after Stanley left, as it was said in 1959, 1960, I came back to my native country, Italy, and there I get other people equally important in my life, as their name is Professor Pietro Calissano, who is following me, is the director of the Laboratory of Neurobiology in the Institute of Neurobiology in Italy, and has been tremendous help in the recent development of his work. Luigi Alloy has been 20 years with me and is equally responsible, if not more, at least like I am, for the recent work which we have done. The very last, and then we go into the work, is to the Swedish scientist I owe my thanks, because without their outstanding contribution to the study histochemical and immunohistochemistry, never the NGF would have been blossomed as it did today, particularly for the work which is done in the central nervous system. Though I suppose the Nobel Prize should be shared with all these people, more than I am responsible for this work, which I enjoyed very much. I don't believe I was certainly number one contributor, one of the many. Now we start in a brief account of how this work began and already has been mentioned, and how it's progressing now, and what are the perspectives. The very first slide, which I saw a moment ago, show already a point which is not really the beginning. With Victor Hamburger, we were very much interested in finding out how the periphery act upon the developing nervous systems, and this was done simply by the classical way of extirpation or implantation of additional limb bird, either in amphibia or in the chick embryo. Finally, a former student of Victor, Elmer Bucher, had a bold and ingenious idea to transplant not a limb bird, but rather a very homogeneous and fast-growing tissue, mouse sarcoma and relative. We repeated the experiment. He came to the conclusion that mouse sarcoma work on the central nervous system not because of any special characteristic of the tumor, but because it grows fast and the fibers enter in, and the cells which send fibers into the tumor have facility to invade a larger field, and this is correct, but that ended, as you were by Elmer Bucher, in 1948. Dr. Hamburger and I repeated the experiment, found something different. This experiment I performed in 1951 and gave to me really the idea that the tumor was working in a quite different way from what was been conceived by Elmer Bucher. What you see here is a diagrammatic representation of the viscera of a normal embryo. Here you have, just in a diagrammatic way, the lungs, L, and then you have the primitive kidney, mesonephros, the gonad, the sex organ, and this is a slim chain of prevertebral ganglia, sympathetic ganglia, and the adrenal gland. Now, when you do transplant the tumor, as we did, thank you so much. When you, as you do here, as we did in 1951, transplant the tumor, which grows very fast, the tumor into a 3-day chick embryo grows very fast, and you see it here hatched, and fibers from sensory and sympathetic ganglia enter in large number into the tumor, and the ganglia which give origin to the fiber are larger than the control. The control axis 1 you see here, and here is very much enlarged. This has not been observed before by Bucher. Bucher thought that the effect was only on sensory ganglia. But what seemed to me extraordinary is not only the fibers entering into the tumor, but invading very large, chaotic ways the primitive mesonephros, which normally short-living organ is not innervated at all. Now, to a method which I developed, I knew well from my work with Giuseppe Levi by Cajal technique, silver technique, I saw that a large number of fibers enter in, as you see, in a chaotic way and branch all over the mesonephros. Now, since it would be difficult to explain only as Bucher had proposed by saying the tumor grows fast, and for this reason fibers enter in and the ganglia invading the tumor, sending fibers to the tumor are larger. So this seemed to me to be the result of a soluble factor which had been released by the tumor, invaded, entered into the circulation of the embryo, and for this reason the ganglia become enlarged and send fibers into all organs of the embryo. To prove this, I tried this experiment, very simple one, very old-fashioned, like I am myself. So I put a fragment of the tumor in the chorioallantoid membrane, quite apart from the embryo itself. Many of you have certainly know how the chorioallantoid membrane is wrapped around the embryo, but entirely separated. Only the circulation is shared by the tumor and by the chorioallantoid membrane and by the embryo. Now, even from this position, the tumor was able to produce this extraordinary effect, enlargement, comparing with sympathetic ganglia, and fibers penetrating in all viscera, even entering inside of the blood vessels, which is an extraordinary feature never seen before. Of course, in neuroembryology and embryology, you see many, many things you don't understand, but this seemed to me too interesting just to abandon it and say why it happened. So the second step, and the one which was really the opening of the entire story, was to try to find out the nature of this particular factor, soluble factor, released by the tumor. No biochemist in the world, not even the excellent Stanley Cohen, would have accepted at this point to come to work with me and to try to find out about the nature of this soluble factor if I had only presented him with this kind of experiment. It was too difficult. It takes months to have this result, and the amount is so small he would never have found. So I decided to go to Rio de Janeiro, where I was a guest of Professor Carlos Chagas, now president of the Vatican, academy in Rome, and to try to find a very simple test, biopsy, to find about the nature of this tumor. The following slide shows to you a drawing which I sent to Victor Hamburger in St. Louis when I was full of joy for what I saw. And this is my drawing exactly. He came back to me when I went back and showed to you the effect. Here, in a tissue culture hanging drop technique, I put a fragment of mouse sarcoma, the mouse sarcoma which has its effect in vivo, and nearby a ganglion. The ganglion produced only fibroblasts, no fibers. But when instead I put, as in this case, the sarcoma here and here are two for 24 and 48 hours later. When I put a fragment of mouse sarcoma adjacent to the ganglion, here it was, I'm sorry, I have to say again, this was a fragment of tissue of embryo, which was not a mouse sarcoma, it was a different tissue which had no effect whatsoever. When instead I put in the hanging drop technique tissue culture a fragment of mouse sarcoma, as here and here, the sarcoma occurs have origin to migrating neoplastic cells, and the ganglia, which are adjacent to, producing in 10 hours a very dense halo of fiber, which were directed toward the fragment of tumor. Here you see 48 hours. It was what I called already at the time a typical neurotropic effect. The tumor not only fosters the cell to produce a large number of fibers in a very short time, but also forces the fibers to go in direction of the tumor. I must say that 2 decades later, we really discussed the phenomenon and were sure about this. Now, this was a drawing. I had not a facility of a camera at that time, and I was too happy and excited to wait for receiving a camera. The next showed to you the effect, as we saw when finally we repeated the experiment, had a much larger amount of NGF at our disposal for the discovery that the NGF was not only present in the tumor, but also was in large amount found in mouse salivary gland and snake venom, the made cost particularly to Stanley Cohen. Here you have the experiment which became the classic standard biopsy for studying where and how the NGF is produced, the kind of sources of NGF. Here you have a ganglion, a sensory or sympathetic, just as the same ganglion, which is grown in a medium, minimal essential medium, with no tumor factor in the medium itself. You see that 24 hours later, you have only very few fibroblasts growing out, no fibers grow out from the ganglion, which is here. The ganglion comes from a dechic embryo. But if the ganglion has been cultured for 24 or even less hours into a medium where 10 nanogram of the purifying NGF purified by Stanley Cohen added to, immediately you have this tremendous halo of nerve fibers, which is very similar to the one which in my first drawing in 1952 I showed to Professor Victor Hamburger. So this gave to us two tremendous advantages. At first, a very fast screening to find out what kind of tissue produced the NGF, and it was discovered that, I cannot go into the details, that NGF is produced in large amount, we will come back later to this point, in many sources, in snake venom and in male mouse salivary gland. Second, he gave to Stanley Cohen, and to other biochemists after him, the possibility of a test which was very, very fast, only last 10, 12 hours, and it was possible for him to make experiment biochemical to identify this soluble growth factor, which became known from 1954 up to today, no growth factor. Not only it became known, identified, but eventually was sequenced, and from there came the discovery of the gene, as I will say in a moment. Please, next. Here it is. When we had this new source, I don't go in detail, otherwise I would take the time of Stanley Cohen. When we found this very important, very powerful source of NGF, which was no more the tumor, by the way, has only very small amount of NGF, and you have to grow it for a long time to have very few nanogram, not even milligram of NGF. But when it was found that NGF, for some reason that only 3 months ago, or 6 months ago, we discovered why is there. When we found that the NGF is present in very large amounts in the mouse salivary gland, and Stanley Cohen purified it and gave it to me, I injected into a neonatal rodent and other kind of animals. Here's the effect that you see. To the left, you have the superior cervical ganglion of a control little mouse or rat, just the same, which is rather small. But if for 10 days you gave to the animal every day a little amount in the body of a 10 gamma, not so little, but not so high, 10 gamma of NGF to the animal, you see that this ganglion grew enormously, and the same is true for all sympathetic ganglia, which are para and prevertebral. Is about 9 times larger, and the amount of growth volume increases due to increase in number of cells, increase of size of the cells. For a long time, we thought that it was a mitotic effect, a proliferative effect, beside being an hypertrophic effect, increasing fiber number. But recently, another person who is responsible, not I, for the discovery, found out that the NGF does not act as a mitogen by increasing number of cells, but by impeding the death of the cells which normally occur in early life. That is, more cells are produced than cells that remain. And so when we give NGF, we permitted to all cells which were there to undergo growth because the NGF is provided to them and they can grow. That is normally an overabundance of cells which cannot survive and undergo death, natural death so-called. When you give NGF, the increasing number of cells is not due to proliferative effect. Eventually, however, recently was discovered that indeed NGF may also have a proliferative effect, but I'm not going into this point. At this point, we know that increase in sympathetic ganglia, which is 6 to 9 times the control, is due to prevention of cell death, hypertrophy, and increase in production of nerve fibers. Please, next. The evidence that NGF is not only a splendid nutrient, very important, but is essential to the life of the cells. And this was proved to an experiment quite good. Again, I give the merit, particularly to Stanley Cohen. He produced a specific anti-serum to NGF, and when he was sure that it was quite powerful, he gave to me. I injected the anti-serum to NGF, which had produced by injecting with the Freund adjuvant into rabbit. I injected the anti-serum into neonatal rodents, many different rabbit and other kind of mammals, and this is the result, which we call immunosympathectomy. To the left, you have 2 ganglia, thoracic ganglia, paravertebral ganglia of a 1 month old, whole mount of sympathetic ganglia by control. To the right, you have what is left of the ganglia when you have every day for 3 days, the first day, and then you quit doing it, you injected the anti-serum to NGF. Why this happened? And this proved, and this we immediately realized, it proves the tremendous importance of NGF, not only for survival, but just not only for growing better, but just for surviving. Without NGF, that is, the NGF had been neutralized by the antibody which had been injected, the cells cannot develop. They undergo death, and we studied in detail the process, and we called it immunosympathectomy. Why we called it so? Because the animal which has been treated for only 3 days in the first days of life with the anti-serum, and this makes no harm to the animal, it's entirely lacking any toxic characteristic, the animal will be deprived for life of the sympathetic system. Later on, we found that this is not only true for anti-serum to NGF. You can produce the death of the cells by different technique, by surgical means, I will in a moment mention, and by chemical means, pharmacological. Surgical means, when it was Jen Handy who did this experiment, means simply to make a surgery cut of the postganglionic fibers of the sympathetic ganglia and prevent the ganglia from invading the periphery. Now, when you do this in a very early life of the animal, of the mouse or any kind of rodent, the cells of the ganglia, which are prevented from sending the fibers to the periphery, undergo death. Why is it so? Again, this was a typical example that the periphery is producing a little amount of NGF, which is retrogradally transported to the cell and is essential for the life of the cell. This is surgical axotomy. Almost at the same time, we did in Rome with Dr. Angeletti another experiment. We injected a pharmacological agent which is known as 6-hydroxydopamine, a dopamine derivative. The dopamine derivative, which was discovered by Turner and Tranzer and others, does this kind of effect. It prevents the communication of the postganglionic fibers to the periphery. In adult, this result is the lack of innervation of periphery by the ganglia, because the fibers have been burned, we can say, or at least destroyed as the terminal, and are no more able to reach the periphery, to make synaptic contact with the periphery and convey the nerve impulse. This is the adult, that is effect is 6 to 8 weeks old, long, and then is the established connection. But when you do HIT, you give 6-hydroxydopamine, that is dopamine derivative, to a neonatal animals, the fact that you have impeded the connection of the ganglia with the periphery for a period which is only 3, 4, 5, or some more weeks, is enough to kill the cells, because in cells of a neonatal animal, or for that matter even before being neonatal, the cells which have not received NGF from the periphery, because the fibers do not reach any more the periphery, are doomed to death. NGF is so important that is prevention from reaching the cells is bringing to death. So this we call chemical sympathectomy, and proves that in any way, when you prevent the periphery from communicating to the centers, that is to send NGF to the center, sympathetic cells which are target to NGF, the cells cannot survive because they need badly, hour after hour, this particular substance, which is, in this case, neurospector. Next, please. Here it shows an extraordinary effect which puzzled us a great deal. When we gave 6-hydroxydopamine to neonatal animals, we had this curious effect. Here you have a normal superior whole mount, toluid in blue, of superior sympathetic ganglion. Here you have a sympathetic ganglion of an animal which received anti-serum and is very much reduced, even if not as the one I showed to you before. Here is the superior cervical ganglion of an animal which received the daily NGF and is 5 times larger than the control. But when you give 6-hydroxydopamine, you have a paradoxal effect. The ganglion tremendously larger than the one which received the ganglion of the animal which received NGF, and is very much abnormal as feature. Simply I say you what happened. What it happened is the following. Not only the 6-hydroxydopamine which we injected prevents the fibers from reaching the periphery, but at the same time we gave daily NGF. Now the ganglion is growing in the vacuum. It does not reach and does not receive NGF from the periphery, but receive 1,000 times more by our daily injection. Under normal conditions, the periphery is necessary for the life of the death, but at the same time, we send some message that says stop sending fibers, even when you give NGF. So the NGF is increasing the size of the ganglion, but at the same time, some message which we don't know come from the periphery and say, now we have enough, we cannot grow anymore. When you have separated the ganglion by doing this chemical axotomy from the periphery, the ganglion is living in itself, in its vacuum. The more NGF you give, the more the ganglion is growing and reaches this tremendous size. So this is a paradoxical, interesting effect. Next, please. Here you have a diagrammatic representation of what I've told you. Here is a normal ganglion, superior cervical ganglion, which innervates many organs, among others the iris of the animal. Here you give 6-hydroxydopamine alone, and 6-hydroxydopamine alone bring about shrinking of the ganglion, death of the cells, and no innervation of peripheral organ. When you give NGF, the ganglion is much larger than the normal ganglion, and the periphery is hyperinnervated. But when you give NGF and this 6-hydroxydopamine and NGF, you prevent any connection of the ganglion with the periphery, as you see, but you put the ganglion in a condition which is extraordinary condition. Receive no message, neither receive NGF, nor receive the message, stop sending fibers, and you have this tremendous enlargement of the ganglion. Please, next. Here it is another effect of NGF, which was discovered in 1976 in our laboratory in Rome. At the same time, shortly later, it was confirmed and improved with improved technique by other people in Barrett and Gunderson in the United States. We injected into the brain of a neonatal rodent with a microcapillary NGF into the center to try to find out if noradrenergic centers in the central nervous system were also responsive to NGF as the sympathetic ganglia, as the periphery. We had an effect which we didn't expect at all to have. That is, we found out that the NGF which has been diffusing from the pipette along the medullary level and spinal ganglia brought about a curious effect which is shown in the following slide. Next, please. Here it is my injection, which I made as the bulbar level, and here you have what happened. The NGF diffused in a gradient from the bulbospinal level to the spinal cord and reached through the dorsal and ventral root the sympathetic ganglia. The sympathetic ganglia not only were larger than the contralateral, but fibers emerging from these followed the gradient up to the point where NGF had been injected. So we came to the conclusion that NGF is not only acting as differentiating entrophic factor essential for life, but also act as a neurotropic factor which directs the fibers along the amount of this NGF. Has been proved recently in a probably more rigorous way. Next, please. Here you have two which show it. Here is a baby rodent, a baby rat. Normally you have no fibers from the ganglia entering into the spinal cord or in the brain. But if you injected, as we did, NGF into the bulbospinal level, you see the ganglia are much enlarged of the sympathetic cord, sympathetic paravertebral chain, and beside invading the periphery much more than in the control, some fibers enter, some collateral fibers into the spinal cord and into the bulb following the gradient of NGF. Next, please. Here you have the histofluorescent. I owe it, as I say, to the splendid Hiller-Fat technique, as histofluorescent technique, well known in this country, now a classic method to study the nerve fiber, shows the difference. Here, through the histofluorescent technique, we show what happened. Here is a control, the superior or the thoracic at this point, thoracic sympathetic ganglion, no fibers enter inside of the spinal cord. Just you have these highly fluorescent. But when you injected into the brain the NGF, you see the ganglia, this white mass much larger than these, at the same time, not only the ganglia send fibers to the periphery, but collateral entered through the dorsal root, penetrated inside of the brain, in the way the diagrammatic slide showed to you. And the fibers remain inside as a parasitic system, which normally, we believe, display no function and will disappear as soon as you abandon, you don't inject it anymore. The next, please. And last of these shows the beautiful experiment by Barrett and Gundersen. They showed the same things, that is the neurotropic effect, in a more rigorous way. That is, the sensory fibers, in this case, grown from a sensory ganglion in vitro, you see here with the periphery, with the top of the fibers, is exposed to the action of NGF, which is released from a microcapilla, this is the microcapilla, and the fibers bend towards the origin of the NGF. And if, as this author did, you move, you shift around these microcapillars, fibers will follow passively the direction. And here you have also again a point you don't see too well, but it's enough you can, in a calamite kind of way, the fibers follow the direction of the gradient of NGF, showing therefore the very important neurotropic effect, which may really play an outstanding role in the formation of neuronal circuit. A lot of work has been done on this part. Please, next. Now, after having shown the first part of the work, which went up to 1976, that is 2 decades of hard work and few slides show to it, we decided to explore the action of NGF also on other cell type. Here you have the group of cells which we know now, the type of cells which we know now are responsive to NGF, and we call it NGF target cells. Neural cast derivative, the sympathoadrenal cells. Sympathoadrenal, I will speak of this in another session in a few days. There are the long sympathetic neuron, the short sympathetic neuron. I'm not going to detail about this, would take too much time. Cells of the paraganglia, carotid and abdominal paraganglia. These are cells which have the same origin from neural crest as the sympathetic, but took a different direction. They are small, they look like glandular cells, and they are also known, many of them, as SIF cells, small, highly fluorescent cells. Fine, and these are the SIF cells, these are the cells of the ganglia, adrenal gland. These are the SIF cells, which are present and very similar to these into the ganglia. Finally, as the chromaffin cells, normal and neoplastic, and this we will discuss in a moment in detail. Finally, besides the sympathoadrenal cells, you have the sensory neuron. I mentioned earlier, I didn't go in detail, that not only the sympathetic cells and the adrenal cells originating from neural crest, but sensory neurons also respond to NGF, not as powerful as the sympathoadrenal cells, but also then they're going increasing size, increasing number, for the same reason that death is prevented. Now, the very recent development and the most important, exciting work, which started in 1983 with the work by Turner and many others, was to find out that not only sympathetic cells and these other kind of cells, sensory cells, respond to NGF, but also neurons which play an essential or most important role in higher brain function. And these are the cholinergic neurons, which are found in the corpus striatum, basal forebrain, septum, nucleus diagonal band of Broca. Ever since his first paper in 1983 appeared, a tremendous number of papers proved the very important roles that the NGF plays not only on the noradrenaline and sympathoadrenal cells from the periphery, and we will see also in the centers, but also on cholinergic neurons, which, as I say, are very much under continuous studies, because we know that a lot of devastating disease, Alzheimer, Huntington, and others, are due to dysfunction of cholinergic neurons. I will come on this later. Finally, we tried, and this experiment we did rather recently with Luigi Aloi in Rome, we found out that in the center of lower vertebrate, particularly in this case we used the Xenopus levis tadpoles, we have a large number of cells, not only the cholinergic, but also the adrenergic, the indolaminergic, the peptidergic neuron, respond in a magnificent way into the Xenopus tadpole, as well as in post-metamorphic stages. And a point which I will touch a moment, because it's very important, we found it was the first matter of chance, later on it became an object of study, we found the cells which belong to the immune system, the mast cells, are responsive to NGF to the same extent as the sympathetic sensory and the central nervous system. Later on I will show you what we mean for this response. Please, next. These are the PC12 cells, the one which first brought to attention the response of chromatin cells to NGF. And this is work by Lloyd Green and Tischer in the United States in 1976. These are cells which derive from a pheochromocytoma, which is a malignant tumor originated from chromatin cells. These cells are very malignant tumor in rat, and they grow fast, and there is a typical aspect of glandular cells, but being neoplastic, they undergo very vivacious proliferation in vitro, and they can grow forever, and you can transplant in tissue culture as long as you want. But if you add to the medium of one particular clone, which became known as PC12 cells, if you add NGF, these cells are channeled phenotypically, not towards the glandular and neoplastic type, but they become similar, almost identical to sympathetic cells. They do not undergo any more division. They have all the features, morphological, electrophysiological, biochemical of sympathetic cells. They stop dividing, and they grow a large number of nerve fibers, which continue to grow as long as you give in the medium NGF. If you stop giving NGF, the cells revert to the primitive condition. This system has been invaluable in giving a model for studying the mechanism of action of NGF. I cannot, in the time which I still have, go in detail, but it has been the model that ever since in 1976 was introduced, offers the possibility to biophysicists, Professor Calzano here and many others, to study in detail what is the way how the cells, through receptor, capture the NGF, how the NGF is brought inside of the cells, and how it works. Still, we cannot say that all the mechanism of action has been discovered, but without this model, it would have been very difficult, because neither sympathetic nor sensory cells are good models for studying the mechanism of action. All of you know that nerve cells do not divide under normal conditions, so the mechanism of action would be very difficult to study in a system like nerve cells. It became instead very easy when the PC12 clone studied by Tischer and Green became available, and people could study in detail all the aspects of this phenomenon, our cells, which is glandular cells, become a sympathetic-like cells, and we can revert to the primitive condition, and it was, as I say, the opening really of the study of molecular biology applied to the NGF. Next, please. In the same time, almost at the same time, in our laboratory, Luigi Alò and I decided to study the effect of NGF not only on neoplastic cells like the PC12 clone cells, but on normal immature chromaffin cells. And here is the result, as you see. Later on, we obtained even more dramatic effect. Here is the chromaffin gland, the adrenal gland of a normal animal. Here is the cortical component. Here is the adrenal component, medullary component made up of chromaffin type of cells. When we inject immediately after birth, in this case NGF, you see that all cells which normally would give origin to glandular chromaffin cells become sympathetic-like and large. In other cases, we have even more dramatic effect and give origin to a large number of nerve fibers which go out and innervate the blood vessels which are adjacent to. But you see a tremendous change in the morphology of the organ because we have channeled, routed the precursor or immature chromaffin cells towards the nervous rather than glandular phenotype. Please, next. Not only the chromaffin cells of the adrenal gland, but all paraganglia, I'm not going into detail, and the carotid body, which has the origin from neural crest. Normally, the carotid body, which is here, is very close to the superior cervical ganglia and also to the nodosum. And it is very important, the feature is the same as of the adrenal cell gland, but the function is different. It is sensitive to pressure into the circulation, blood pressure, and ionic change. When you inject in the neonatal animal or in the fetus, as we are doing now, NGF, all this small paraganglia carotid body, which is here and very small, become channeled and look like a sympathetic gigantic ganglion, which does not display anymore the regular function about the blood pressure and the change, ionic change into the blood, but act like sympathetic type of cells. Please, next. And here you have what was the rather recent discovery that Luigi Alò and I made in 1977, but only now we went on in developing. We studied through the famous ILAP-FAC technique in our neonatal rodent which had received NGF, and we did not expect. This is truly a chance discovery. We thought that, of course, we would find that the animal which received a large amount of NGF would have much larger sympathetic system, and this was true. But besides the increase in sympathetic cells, which are highly fluorescent, this was the reason for the FAC-ILAP study, we found out a curious phenomenon. All the tissue, peripheral tissue, are loaded with small, highly fluorescent cells, which we had no pain in discovering. It was very simple that the cells were not neuronal cells, but were mast cells, which are very fluorescent because of the amount of histamine and highly fluorescent substance dopamine also inside of the cells. Professor Unes here wrote a splendid classic paper some years ago where he said that the mast cells is very similar to sympathetic cells, the same kind of picking up as a precursor and using. So the mast cell, which is today considered as a paraneuron, is in this particular case forced to increase in number tremendously. Here you have the iris of a normal little rodent, 1 month old. Here you have the iris of the rodent. You see the nerve fibers are greatly increased in number, sympathetic fibers. The plexus is much more fluorescent because sympathetic cells are increased in number, the fibers are increased. But at the same time, you have what you don't have in the control, a large number of these masked, highly fluorescent cells. We know that there are mast cells through different techniques, which I'm not showing, through ultrastructural study and metachromatic study and biochemical study. So the problem was raised. Is it due to a secondary effect because we have increased the nerve fibers by injecting NGF? That is, are the cells there only as a secondary effect to increase in amount of adrenergic fibers? We proved that this is not the case because we gave 6-hydroxydopamine and it was no more innervation of the iris. You see here is no fluorescent fibers, and the number of cells was even larger than in the control. Namely, this proves that these mast cells are responsive to the NGF to the same extent, even we can say even more, than the regular sympathetic and sensory cells are truly NGF target cells. Next, please. The study is now advanced. Luigi Alloy produced anti-serum in two ways to NGF through autoimmunization, that is, injecting a neonatal animal with, well, injecting even an adult, a 2 months old female with NGF, and the animal became autoimmunized against his own NGF, which is normal in circulation. And this is the result. When you study, a few months later, where the animal is autoimmunized, the mast cells in the peritoneum is a normal case. That is, the mast cells which are found are regular, filled with granules, just the typical kind of image you would have when you look at the peritoneal mast cells. But if you now look at the peritoneal cells of an animal which has been autoimmunized, the number of mast cells is much less than in the control, and the cells are devastated, degenerated, you have granules all over. This means that the animal has been autoimmunized, and the lack of NGF produces not only an effect on the sympathetic and sensory system, for that matter, but also act on the mast cells, bringing about this very devastating effect, a decreasing number and degranulation of the cells. So this is an additional evidence that NGF is important not only for sympathetic, sensory, and chromaffin cells, but also for these cells. We have here a linkage between the nervous system and the immune system, which is under intense investigation in our different laboratory. Next. We have now studied in Rome the effect of NGF on the precursor of mast cells, taking a small fragment of a spleen of a normal neonatal rodent and growing this in tissue culture and studying the colony which came about. Now, when you add it to the medium NGF, you have that practically all cells of this colony are undergoing channeled toward the mast cell type. Normal in the normal condition, a number of mast cells is found also in control, but here is extraordinary the number of mast cells. So the NGF act on mast cell precursor. We still don't know if on one particular type of precursor or all the cells. Many people are studying this phenomenon and studied also the NGF effect on leukocyte proliferation and lymphocyte. So it is now accepted in the literature that NGF act on immune system, and this may give us a clue about the study of neuromodulation and neuroimmune system. Next, please. I'm sorry, I'm going to go very fast because I do believe I am almost at the end. How much time is left? 5, thank you. Now, here you have, I have a friend who was just requested to tell me when I should stop. But I have still some things to say, please. Be patient for 5 more minutes, or 6 perhaps. Now, if you don't mind. Now, here you have an experiment performed by Luigi Alloy that we were wondering how the mast cells respond to NGF. And here are mast cells which are easily recognized because of metachromasia. And if you give labeled NGF, they are highly labeled. Those have a receptor, as they should have, for NGF exactly the same way or the same amount, density as sympathetic cells. We are now studying the mechanism of action of NGF, how the spleen cells, what kind of spleen cells respond to NGF, and if other cells of the immune system, as we do believe, respond to it. So the NGF is now acting not only on the mast cells, sympathetic, sensory, and chromaffin cells, but on the cells. Now, I very rapidly show to you the effect of NGF on the central nervous system of Xenopus levis and of rodent. Next, please. We have no time, very fast. Oh la la, this, I'm afraid we should stop. I cannot go on this. This is the sequence. In 1970, Angeletti, Rute, and Raprashaw were the first to find the amino acid sequence of the NGF protein molecule and found how it is. But I have no time to go on this. Next, please. Here is very recent work by the group of Scott and Ulrich, 1983, is going out now, is a product of the gene. The neurogrowth factor is a part of a pre-pro NGF. is only 118 amino acids of a longer chain of amino acids. We don't know what is the function of this part. We know through the molecular study of molecular engineering that the inner growth factor is also the second chromosome or a quark in the short arm of chromosome 2. Next, please. I want to remain in my own field. I would not have time to cover it all. Now, here is the action of NGF in the central nervous system of Xenopus, tadpole, stage 42-44. Here you have the pontomesencephalic indolaminergic cells, highly fluorescent, obviously actinic, paraventricular cells, close to the third ventricle, postinfundibular cells, and here is the size of this particular cells. When you give to a tadpole stage 46-48 NGF, you have mass NGF, you have a tremendous number of cells, much more fluorescent here, I cannot show too well, of indolaminergic cells, pontomesencephalic, noradrenergic, paraventricular, postinfundibular, and you have these cells are not only more fluorescent and larger, but each cell individual undergo quite a dramatic increase as compared to cis. But it's not only the noradrenergic and the indolaminergic cells which respond to, also cells which we don't know, now we believe we know what kind of transmitter release the Bowner cells. Next, please. Here you have the famous Bowner cells. Nobody knew what was this transmitter. We know now that it's substance P. Under normal conditions, this is a very large cell, as a pointing level. Here is a very much increased cells with a large number of dendrites following injection in this tadpole of NGF for a few days. Next, please. And here you have the effect of NGF on other kind of peptidergic neuron. That is, we found that the peptidergic neuron in this particular species, we still don't know about rodent, respond to NGF equally, even more than noradrenergic and cholinergic neuron. Here you have, as the mesencephalic level, a substance P. You have the very few cells fluorescent in the mesencephalic level. Here is, following injection, substance P is much increased. Here is a somatostatin, pontomesencephalic level, control and experiment. So we have not only that cells which we don't know what function is, what kind of chemical transmitter they have. Now we believe it's substance P, but we know that all the cells in the center of Xenopus and lower vertebrae respond in a tremendous way, even more than a higher vertebrate, to NGF. Next, please. And here you have a long frontal section by Luigi Eloy. Substance P is the spinal cord and brain level of a control. Substance P highly fluorescent in a tiny tadpole which received for 4 to 5 days NGF. So all substance P you see is not only the brain but all along the body. Next, please. And here we have very time to say, even if I like this particular chapter very much, but there's very little time left from what I was told. Perhaps there's only 1 minute left. So this is a discovery which seems to me quite fascinating. We found out that cells which are surrounding the subventricle and respond to the circumventricular organ respond in a magnificent way to NGF, and probably are the first to receive the message from outside. These cells are called very curious cells, either tiny size that is transformed glia cells or primitive sensory neuron. Nobody really recognized one from the other. These cells, under normal condition, are located in some area around the subventricle here and are known as window in the brain. Being the object of a large amount of study, is considered that a message or chemical agent captured from outside, from the brain, enter through this window in the brain into the hypothalamic level and act upon the hypothalamus, bringing about the effect that the hypothalamus is releasing a hypothesis and starting the chain of events which are known as neuron docking differentiation. Next, please. Here are the kind of cells which belong tiny side, transformed or modify glia cells, and at the same time, according to us, I would believe, are primitive kind of sensory cerebral neuron, very primitive neuron rather than glia. Next, please. And here you have, with the Cajal technique, under normal conditions, it's our ventricle, these cells just are bulging inside of the ventricle, are very, very small, as you see here, and barely starting to differentiate. When you give NGF, next, these cells are terribly increased number by the thousand and virgin side. So the cells capture the NGF and bring the message through a particular way to the hypothalamus and starts the chain of reaction that is neuroendocrine differentiation. Next, please. Here it is NGF, which has been labeled NGF with iodine 125. Here is its third ventricle in a control. Here is its third ventricle of a little rodent, in this case is a rodent, which was receiving NGF. You see a large number of cells not present here, which are loaded with NGF, and eventually an effect which we described and we believe are related to SEES is an action on hypothalamic centers. And the experiment we performed was the following. We injected in prenatal, Luigi Alloy, not I, we injected anti-serum to NGF, and the pups born. This is the male which received NGF. The NGF, I'm sorry, did I? Is aggravating the lack of time. Well, I'm sorry. Well, the animals were injected individually through cesarean cut incision of the abdominal wall, and Luigi injected each one with anti-serum to NGF. The litter, the pup were born normal. They looked exactly as the control. But after 2 weeks, many of them, 60%, became stumped and eventually died for starvation. The one who survived, next, show to you. This is a pup, 1 month old. Here is a pup which received in prenatal life through injection, intramuscular injection by Luigi Alloy, anti-serum to NGF. You see how smaller it is in this and how deficient is the position, wrong position of the leg. The sensory system is altered, but more than all, it shows epithetic behavior and very severe neuroendocrine syndrome. Next, please. Here you have the normal thyroid of a control, the very much reduced thyroid of an animal which has been treated with this. I'm afraid that I should stop here and not tell you what you can read, however, in one of the last notes of TNA.
That is so difficult.
It's the fault of the system, not really mine. Well, the very last part, which I'm not able to cover without damaging what is most important and is taking away time from my friend Stanley Cohen, what we found out, and I hope some of you will read, is that we discovered why the NGF is present in the mouse salivary gland. This has been for a long time really a pestle. Why food? The mouse salivary gland has so much tremendous amount at the order of magnitude more NGF than any other organ. And the pestis was even more, the animal, the mouse is a very, very aggressive. No, I'm behaving. Well, the animal, the mouse is a very, very aggressive, a kind of mammoth, interspecific kind of aggression. And each male survived by killing other males. The female take no part in this aggression. So we decided to study, to explore the action of aggression on the NGF in the salivary gland. It's very easy to make a mouse aggressive. It's also easy to make a man aggressive, but it's even more easy to make a mouse aggressive by just isolating the mice and putting them together later. When they see each other after a week, after 4 weeks of isolation, they start cutting each other's throat and they try to destroy the other one. If you take the blood from the retroorbital plexus, when the animal has gone many feet, many of aggression, you will see that the blood of this animal is loaded with NGF. And this, we do believe, is in order to help or to increase its capacity to stand stress. Only this kind of stress, interspecific fighting, any other kind of stress would do nothing at all. So it was found out that a large amount of NGF is released into the blood only under this condition. So the animal becomes very, very aggressive. How the order comes and from where it comes, recent work by Luigi Alloy, very impressive, gave a very impressive evidence how it comes. It comes from production of NGF in the hypothalamic centers. I am not able to show to you, it would take too much time, but we have slides proving that animals which are becoming very, very aggressive just because they face each other after isolation, and they are just, as I say, fighting to each other, not only have the blood loaded with NGF, but the hypothalamic centers at this moment become, as a large number of cells which have, in the normal condition, when there are quite very little amount of NGF, the cells are producing a large amount of NGF. At the same time, in the laboratory, I believe, of Ulrich, at the same time as we did this experiment, it was found that messenger RNA is present in large amount in the cells. So we do believe that these cells are normally releasing NGF, but far in more amount, larger amount, when they are under this kind of intraspecific aggression. How from here we have the result of the release into the blood from the salivary gland, still we have not found out. But we know that the adrenal gland undergo very much increase in size, is particularly the adrenal, but also the cortical part, when we inject exogenous NGF and also when endogenous NGF is produced. So we are now following this line to find out how from the hypothalamic centers the order is sent to salivary gland to release NGF, how this NGF will in turn increase the capacity of defense of the aggression, capacity of defense of the neuroendocrine system, and in particular the activity of the adrenal gland. I'm sorry for the time I took to use, Stan. Thank you so much.